Okay, here's the second lecture for Chapter 6 till the end. Right, so we're going to be talking more about relationships between organisms in a later chapter, but this introduces you to the concept of relationships. So bacteria have a lot of relationships with other organisms. Some bacteria have relationships with other bacteria, cooperative or predatory. Some bacteria have relationships with protozoa. Bacteria can have relationships with animals, as in normal flora in humans. Protozoa have relationships with animals. For example, the picture up at the top right, you're looking at termites. And inside of the termite gut is a protozoan. So the termite is the host that benefits from the protozoan. The protozoan benefits from living inside the termite. You can see the protective benefit of the termite being the host for the protozoan. You may not as easily see the benefit to the protozoan. Protozoan gets food. The termite gets the result of the breakdown of wood done by the protozoan. The protozoan makes an enzyme cellulase, which is used to break down cellulose in wood. That enzyme is not made by a termite. So without that little symbiotic organism in its gut, termites could not eat wood. So that's a synergistic relationship. Actually, it's symbiotic. They are dependent on each other. Some relations are antagonistic. We'll talk again more about this when we get into flora in a later chapter. To take it one step further, microorganisms live in relationships with other microorganisms. You see a shot of this over on the right. Over here, you are looking at microorganisms growing on the surface of the teeth. The tooth surface is this lighter blue, and you can see a variety of bacteria all over the place here, kind of growing on top of each other. This is called a biofilm. A biofilm is a conglomerate of microorganisms that start out small, and by chemical factors that are released to the microorganisms, that kind of gives a shout out to other microorganisms. Other cells are drawn to the area and they stick to each other, creating a film, a layer of microorganisms, different kinds of microorganisms even, on the surface of the object. You could have fungi and bacteria living together. You can have bacteria of different species. It doesn't have to be even the same type of organism. I mentioned that they have a kind of a molecular shout out that draws other organisms to the area. That's called quorum sensing. These are chemical attractants that serve to be a draw to other microorganisms to come to the area. Why are biofilms important? Well, let me point out some important points unrelated to medicine first. They do a lot of damage. They congregate as layers in machinery, in pipes. They erode and corrode equipment. They reduce the diameter of the pipe because of the accumulation of all the microorganisms. They produce chemicals which may be toxic or may have a detrimental effect on things like food, in the case of food processing. From the medical standpoint, of course, on your teeth, I've just shown you a biofilm over here. They are responsible for plaque, which damages your teeth and eventually will cause erosion. If they get into the blood and into the body in a protected area, they overgrow in areas where there may be implants like hip implants, knee implants. They very often will accumulate in areas where there may be a stent in a blood vessel. So this contributes then to the possible injury to the person who got these objects, with the idea that, of course, they wouldn't have any kind of microorganisms growing on them. So they can, in effect, cause the implant to fail eventually. Here's a look at how biofilms occur. Here's the beginning of a couple of organisms that attach, the release of chemicals that draws other organisms to the area. Sometimes bunches of organisms will break away and go off to get involved somewhere else on another area. But eventually, as you can see, they cause an obstruction in the implant or in the pipe or in the whatever. The layers become thicker and thicker. It's very hard to get rid of them by antibiotics, with antibiotics or with antimicrobials. So they're very, very, very big in medicine, and you'll find a lot of information on the internet, for example, about them. Okay, so let's go over to bacterial numbers. I like this picture because it shows you a mixed culture of bacteria. And there are lots. This is obviously not pure, and you should be able to tell that because you've got different colors of colonies, different sizes of colonies, different consistencies of colonies. A colony, let's look at this guy right here. This is a bacterial colony right there. It started with one bacterial cell that ended up on the agar. And that one cell divided into daughter cells, more daughter cells, more daughter cells, more daughter cells. And in a very short amount of time, because of the logarithmic or exponential growth, you have a bunch of daughter cells sitting on top of the original mother cell, creating a mass on an agar plate, and that is called a bacterial colony. A bacterial colony is a clone from the original mother cell. This was the original mother cell right there. They are all of the same species. Now, that colony looks very different from this colony, which looks very different from this little pink colony, which looks very different from this yellow colony, this dark, actually purple colony, very different from the orange colony here, clear, transparent colony. And then you've got some other colonies that are also white, but they look very different. So you've got at least 10 different species of bacteria growing on this agar plate. This is a mixed culture. Now, if you're doing identifications of species from this specimen, maybe it's a wound specimen, it's not, but let's say it is, you have to separate out all these species. You can't run identification tests on 10, 15 organisms all growing together. You can't run antibiotic sensitivities on them when they're all growing together. They have to be separated. Now, there are different ways to separate them. In lab, we're going to be doing some of these methods. We're going to be using streak plates. In a streak plate, you can see a couple of examples up here at the top. Here's the loop, for example, with the original specimen bacteria. It is streaked back and forth on a plate. Then it is spread on the plate, flaming in between, spread more, and then you can see it's spread down here. Now, what happens is you get a dilution effect. This is your most concentrated up here, less. As you go around the dish, you've got fewer and fewer and fewer cells. So eventually, you end up seeing individual colonies right here. And you can see the same thing over here on this plate. Now, this plate looks totally different. It is a pour plate. You can identify it as different because A, you don't see the streak lines. You can see some of those streak lines right there. But the colonies are all spread out equally all over the plate. And you don't see that up here. Here, there's no way to count the colonies. There are large numbers of colonies here, few colonies down here. This is equal distribution. So this is a pore plate. The specimen is put into liquefied agar, mixed. It solidifies. The bacterial cells are locked inside the agar. They undergo growth, divide into daughter cells. You get colonies. Some of the colonies are on the top of the agar. Some of the colonies are actually inside of the agar. You can see the small ones are locked inside. Now, there's also spread plates. A spread plate is the same thing as a pore plate, except that the specimen is only put on the surface of the agar plate instead of being poured into the agar. Now, as I mentioned, with these two streak plates up here, you have to dilute out the sample, and as you streak around the plate after flaming, you get a dilution of cells, which gives you fewer colonies. You have to do something similar to that in a pore or a spread plate. You have to dilute out the specimen. And eventually then you make plates from these specimens. You have most amount of cells here, less even there, and least right there. So 3 different ways to do isolations. Okay, types of media, defined versus complex. Defined, also sometimes called chemically defined or synthetic. So if you look at this, let's see, let me go back. If you look at this recipe right here, there are 3 recipes. Here is a synthetic medium, defined medium for E. coli. Now, look at all the chemicals. Every single chemical is listed. Every amount of every chemical is listed. I could give this recipe to everybody in class, and well, assuming you're able to weigh out it accurately and all that, everybody should end up with the exact same medium. Everything is known. Every amount is known. There is nothing unknown in this medium. So this is a synthetic or chemically defined medium. This is a different synthetic or chemically defined medium, is to grow a different kind of organism. The nice thing about defined media is that depending on what you want to grow, what type of organism, you can add nutrients, vitamins, growth factors that provide for really nice growth of the particular organism you want to find. Now, look at this medium over here. It's short, but that's not a factor here. You can see there's a pH buffer right there, and there's water, and there's glucose, so you know what all those are, obviously. But look at this right here, yeast extract. What they do is they take a bunch of yeast, kill them, sediment them, and then take the solidified yeast material. And they're adding that as a growth supplement. Now, what is inside of a yeast? Well, I'm sure they know all the chromosome information on the yeast. They don't really tell us what kind of yeast. There's probably some protein, amino acids, glucose, things like that mixed in. But this is an unknown factor. I mean, if we knew exactly what was inside of yeast, we could make it from scratch instead of growing it to add for wine production or production of alcohol. The other thing that's an unknown here is peptone. Now, this is a protein material. It's comparable to what you have when you cook a roast, for example, or you bake a chicken and you get juice at the bottom of the pan. That's basically a variation of peptone. And you know it's high protein. It's got some other stuff in lipids, stuff like that, but you don't really know what the analysis is of that material. So this is a complex medium because there are at least 2 things right here that are unknowns. Something in the ingredient of the recipe is an unknown. Okay, so staying with media. Categories of media based on function. There are 4. So let's go with the easy. Let's start with reduced media. No oxygen. Why would you want to use a reduced medium? If you're trying to grow an anaerobe. Okay, enrichment medium. You want to grow organism X. And let's say organism, well, let me give you an example. You want to grow an organism that you could use to degrade petroleum. So, if there's an oil spill, you could release this bacterium. It would degrade while it's undergoing respiration, cell respiration. It would degrade the petroleum product, and in effect you would have been able to sop up the oil spill because the organism is using the material that is the spill. So how would you find this type of organism? Well, you could get it from water, maybe soil. What would you grow it on? Well, you'd probably want to put together a medium that has petroleum in it. If the organisms don't grow on the petroleum medium, then you don't want those organisms because obviously they can't use it. You're looking for things that would grow on the petroleum medium that would show you that it's able to use petroleum as an energy source. That's an enrichment medium. You add something to the medium that favors the growth of a particular organism you're looking for. Okay, selective medium. Kind of the opposite of enrichment. In a selective medium, you add some sort of inhibitory agent. For example, let's say you're trying to find halophilic or halotolerant microorganisms in water. You might make a salt medium, a very high salt medium, because halotolerant and halophilic bacteria need or can live in high salt. Those organisms that are not able to do that would not grow. You don't care about them. You're looking for the salt lovers. So you're using salt as your inhibitory agent. That's selective. Okay, that leaves us with differential. In a differential medium, something's been added to the medium, usually some nutrient and a pH indicator, that causes a change in the medium in that the colonies will turn different color depending on what they're doing. So if you look at the picture right here, this is McConkey agar. Both of these are McConkey agar right here. But this has got E. coli growing on it, and this has got salmonella growing on it. Now it started out the same color, but what's happened is E. coli is able to use lactose sugar. And as it's utilizing it, it makes acid, and the acid causes the pH to change in the medium, and there's a pH indicator added to McConkey agar, and it causes the colonies to turn a red, pink, maroon colony color. You can actually see it in more detail down here. Now the organisms that don't use lactose, the guy on the left, they're growing, they're not changing the pH of the medium because they're not using the sugar. The colony color does not change, and that's this guy right here. So you can differentiate between two different organisms on this medium. Here they are mixed together actually on a plate. You can see that this guy is a lactose user, and this guy does not use lactose. Two totally different species. Okay. So, what do you do when you want to keep cultures? You don't want them to grow, you want to stop them from growing, you don't want to kill them, you just want to reduce their growth. Well, you can put them in the refrigerator, which we're going to be doing in lab. You can put them in a -80 deep freeze, which we also do in lab. You can also freeze dry them, that's called lyophilization. And the vaccine that you see over here, this is the shingles vaccine. You probably can't see in the bottle, though, there is a little pellet, a freeze-dried pellet. Sterile water or saline is added to it, mixed up, and you've got vaccine to inject. It can be sent in the mail as a freeze-dried solution, and it's very safe. As you might already have thought, the mail service doesn't like growing organisms to be sent through the mail. Okay. So let's talk about growth of microorganisms. basically we're going to be looking at two different kinds of growth. There's binary fission, which is what the bacteria do, most of them anyway, where one cell, here's your mother cell, splits into two. The cell has to get bigger. You can see that the cell got larger so that when it divides in two, it's got two cells which now start out small and they get bigger in order to do the same thing. This is not mitosis. It's kind of, your end result is the same as mitosis, but you have no centrioles and you have no spindle fibers. And remember that bacteria only have one chromosome. The mechanics are not the same. Here's a good shot of a bacterial colony, by the way, over there with all the daughter cells on top. Now, binary fission is asexual. You start out with one cell, and that one cell divides into daughter cells. There's no other cell, there's no male cell, female cell, anything like that that came together. Asexual. Another asexual reproductive mechanism is budding. You have a mother cell, and a duplicate DNA copy gets kind of pushed over into one. Here's the rest of the DNA over here. And this begins to kind of, I'll say, pooch. That's not exactly a scientific term, but anyway, a bud forms, and this budding cell gets bigger and bigger. It's got its own DNA copy over here. The mother cell still has hers. And then you can have a bud on top of this, or you can have multiple buds. Sometimes these buds break off and go off on their own. Sometimes they stay attached, and that's what you see over here. And this is the way the yeast asexually reproduce. We'll talk about sexual reproduction in a later chapter. So here you're looking at the growth of a bacterial cell, your starting cell, with one. And in its optimal growth phase, you're going to have 2 cells, 4 cells, 8 cells, 16, 32, 64, blah, 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 over time. So you can see that it starts out kind of slow, and then as the numbers mount, it just begins to skyrocket. And if you look at the numbers over here, in 7 hours, you can have over 2 million cells. You can inoculate an organism on a plate on a Monday and see colonies on a Tuesday. We do it all the time. We streak our plates on a Monday, looking at them on a Wednesday. Inoculate them on a Tuesday, look at them on a Thursday. But you could actually come in 24 hours later and you'd have a perfectly good plate to look at. Notice that the doubling time that if you look at these numbers over here, the cells are doubling every 20 minutes. That's called generation time. The time it takes for the cell population to double. 20 minutes is not weird. E. coli does that in 20 minutes. It doubles in 20 minutes when everything is favorable for its growth. Okay, so here we are at growth. Okay, so 2 graphs at the bottom. Here is time. There's no time put on here, but this could be 10 minutes, 20 minutes, 30 minutes, or, get my thing here, this could be 1 hour, 2 hours, 3 hours. It doesn't even matter what it is. Here is your starting number, and let's say that let's say this is minutes actually. I don't know what I want to use here. We'll say it's hours, actually. We'll make this hours. So let's say that we have 5 cells right here. Okay, if you look over at the y-axis, that's about a 5. And then right here, see if I can write on that, we've doubled, right? There's the 10. And then over here we've doubled again right there. The line has passed over the 20 line. And here we have doubled again, and we're off the chart now. So it starts out kind of slow, but then notice that the line becomes a straight line. This is when the conditions are optimal for its growth. That's called the log phase of growth. And you can actually determine the time it's taking to double using this graph. If this is hours, and let's say right here is where it crosses over the 20 cell line, and let's say that that is, well, we'll just start there. Look at the 40 cell line right there. Okay, so if we go down here to this line, I kind of wavered a little bit over there, and we go down here to this line. Remember, each one of these is an hour. That's right about an hour that it took to double. That would be the generation time, 1 hour to double. Now, as you saw before on the previous page, bacteria double. So again, when we get up here to like here's 65 cells right there, we're going to be at 130 cells next. We're going to be at 260 cells after that. And our graph is just going to skyrocket like that. This poses a problem when you're doing graphing because you'd have to have really long graph paper. Makes it impossible to do it. So usually, either when you're charting bacterial numbers, you either use graph paper, which is what they are using here. This is special graph paper. Look at the divisions of the lines here. They are uneven compared to the ones over here on the left. Or you can use regular graph paper, but you use logarithmic numbers, which is the usual thing you do. And when you use log numbers or you use log paper, you get a straight line, which is way easier to read and way easier to predict numbers on. Okay, so the formula that you're looking at, this is an easy little formula to use to determine the n number of cells given a starting number of cells. So B is your starting number of cells. Let's say we start with 100 cells. And we want to know, after 3 generations, 3 doublings, how many cells are we going to have? Well, the n, the small n right here, is the number of generations. So times 2 to the n would be 3, 2 to the third is 8, multiplied by 100, you've got 800 cells. So after 3 generations, that is, this 800 would be Z, that would be your end, you would have 800 cells. We'll do some examples of this work in class. Okay, so I've taken you through generation time and generations. So that what you're looking at here is a population growth curve. So you take a loop full of bacteria. Here's a tube of sterile broth, nothing in it. Here's a loop full of bacteria, bacterial cells. You stick it in here. You put it into 37 degrees. So what will happen? Here's our start. You can see that pretty much nothing happens. It's called lag phase. There are some cells that are beginning to grow, and they're dividing, but not much. It's negligible. And the reason is that the cells haven't adjusted to the new nutrient. Maybe we took this inoculum from a sucrose broth, and the new medium we put them into is a lactose broth. So they have to actually make an enzyme to break down lactose because they were using a totally different sugar in the other medium. So that takes some time to read some genes to get the enzymes to break down lactose, or maybe the temperature is different. Maybe the temperature of this medium was 25 degrees, where that culture came from, and this new medium has just come out of the refrigerator and it's cold and it's at 10 degrees. So there may be a temperature adjustment. For whatever reason, it's called lag phase. Then everything now is perfect at this point in time from here to here. Temperature optimal. It's got all the enzymes needed to break down the nutrient. pH is being buffered. Everything is optimal for growth, and this is where you get the doubling. And this is when you can calculate generation time. But now things are not so good when we get up here to the stationary phase, because now the pH is beginning to change and become more acidic, which is not good for the cells. We're running out of room. Oxygen levels are dropping. The environment is just not as good. And so you get kind of a reduction of growth rate. Some cells are still dividing. Some cells are actually starting to die, but you get kind of a plateau. And then eventually they go into death phase. They will go into death phase. Okay. Here's a look at some different kinds of growth curves of different bacteria. If you look at this one right here, that is Fusobacterium naviform, and it has a pretty nice growth curve comparable to the one I just showed you. The lag phase is over in this area. It's kind of hidden by all the other colors. Then here's your log phase here, and I already have the arrow on the stationary phase, and they haven't, this is 250 minutes, 275 minutes, about 4.5, 5 hours. It's not enough time for them to have gone into death phase yet. But then, if you look at something like this green, which is Bacillus polymixa, it's got a very short lag phase, then it goes into a log phase and kind of changes a little bit. Stationary phase, real quick, goes into a death phase. So again, there's a lot of variation in these growth curves. Okay, so let's count bacteria. How do you do it? You can do it directly. Very accurate. So, you can do it by a viable plate count. That's a standard plate count. I just showed you what that looked like a couple of pages back, talking about pour plates and spread plates. That's called a viable plate count. Only the organisms that are alive will grow on the agar plates or in the agar plates. You can count the number of colonies. You can multiply it by the dilution, and assuming your math is good, you can get a very good calculation, accurate calculation of number of bacterial cells per gram of sample, per mil of sample, whatever your sample is. And you can do viable plate counts on anything from raw hamburger meat to sponge that was ground up in a vortex in a mixer, to counts on water, to counts on milk, to counts on beer, to counts on soil. We do soil counts also using the same. So anybody can do this. It's got a lot of dilution tubes and things and pipetting, but it's easy to do. It's very low tech. Membrane filtration. You're looking at that down here. This is the apparatus. Inside of this, you can see a little yellow thing right up here. This is a filter. It's got holes. Your sample, you have to use a water sample on this. Usually they use membrane filtration to do water counts on streams or lakes or whatever. Your sample goes through the filter, collects down here. The pores on the membrane are smaller than the bacteria. The bacteria get caught on the top of the filter. You take the filter off, you put it on an agar plate, you incubate it. Here is an actual filter on a plate. These little dark splotches are bacterial colonies. So there's agar underneath here. The bacterial colonies grow on top of the filter, just like they would grow without the filter. Here's a look at the filter. It's in gray. The bacterial cells are caught on top. As you can see, the diameter of the cells is bigger than the holes of the filter. Membrane filtration. Microscopic counts. You see that over here on the far right. Okay. So you got a microscope slide over here. You've got a special slide. It's got a grid on it. You can see the grid is etched into the microscope slide. You put your sample on here. Here's what it actually looks like under a microscope. You have a counter in your hand. You actually count the number of cells that you see. Problem here is that these could all be dead. We could have autoclaved or sterilized the sample and have lots of cells, but they're not viable. They're not alive. Because you're not growing them, you're just counting them. So that's a problem with this. It's good for even doing counts of dead cells, but again, you can't differentiate dead from alive. Whereas with these first two methods, remember that the bacteria have to grow, and only live cells grow. Then we've got electronic counter. That's what this is for. It's picking up charges as the solution goes through the counter. Again, you could have dead cells, live cells. Doesn't tell you whether they're alive. And then another one that's used for water is most probable number. This is more of a guesstimate a close, accurate guesstimate, but still a guesstimate, used for water samples. Well, trying to get the bar there. Okay, then indirect methods. These are methods that don't actually give you a number. Okay, so in lab we're going to be doing spectroscopy. You've done it before in general biology. Take a look at these two tubes. Here is a sterile tube of broth. Here is a bacterial culture growing. And notice that the culture creates a cloudiness in the broth. The cloudier it is, the more cells there are. Whereas the clearer the solution, the less cells there are. Now, this can be useful if you put the cultures, the specimens, into a spectrophotometer. Spectrophotometer has a lamp in it. The light rays go through the solution. So if you put this tube right over here, that's that tube right there, and there's no growth in there, it's a sterile tube of broth, you get 100% transmission of light. You shine a light through this left-hand tube, there is nothing in the tube to stop the light from going through. So all of the light goes through. You get 100% transmission, 0% absorbance. Notice absorbance is the outer scale. Transmission is the inner scale. Now, the culture on the right, the cloudy one, look at this tube down here. The bacterial cells are blocking light. So what's going to happen is you get less transmittance of light, and you get more absorbance of light because the light is absorbed by the cells floating around in the solution. So this gives you absorbance values that then can be useful. Now again, this machine doesn't tell you anything about numbers of cells, although you can use these values to give you an idea, a guesstimate of cells with a graph, which we will do in lab. You can also measure metabolic activity, which will give you an indication whether there are cells there or not. And you can also do dry weight. But again, these are indirect methods. They don't actually give you counts.